So now we're going to use our different method for data collection. And in this particular case, rather than collect specific temperature, pH, sunlight intensity data points, we're now going to look at species diversity as compared with the forest and the open field. So here we're going to use the transect method, where we're going to have a 6-foot transect, and we're going to look at every 2 feet of distance from the border of the open field and the forest. And we're going to make note of the different plant species that we encounter. Now, it's not important that you be able to identify the different species. We just want to see how many different plant things we see within our transect area. So now you're going to see that what the students are going to do is they're going to first start at the border of the two areas, and they're going to clip a leaf from every plant that looks different. Once you've identified all the different plants that you see in your transect area, we're now going to move 2 feet out into the open field. We're going to survey that area again to see, do we see the same plants or is there a different one that we see? And if we do find a different one, we're now going to take a sample of that different one. And we continue to do that until we now can move to our next 2 feet interval and repeat the same thing. There may come a point when you don't see any different plants in the different intervals as you move further out into the open field. And that's okay, because again, we're looking at the species diversity. Once we have moved to our full transect length, now we want to preserve those species so that we now can later look at them and see if there's any correlation with the data we took previously and the number of species that we see in our transect area. So now we see that we have collected some of our samples, and as you're looking at these samples, you will see that we have 1, 2, 3, 4, 5 different species that we have collected in the open field. And we're basing the differences on the leaves of the plants that we have taken. So why are we focusing on the plants and not the other living organisms in the ecosystem? Well, it's important to remember that often the variety and number of plants in an ecosystem will often dictate the other type of animal-based organisms, fungal-based organisms, and bacterial-based organisms that we see in our ecosystems. So because plants are such an important foundation to ecosystems, it sometimes is important to look at their diversity, because that diversity sometimes can tell us more about the health of an ecosystem than looking at things like animal populations. So now we're going to demonstrate the transect method as we move into the forest. So again, we want to start at the same location on the border of the open field and the forest, and we want to move 6 feet into the forest. Once we have set up our transect, we now are going to repeat the same method we did in the open field. So every 2 feet, we're going to collect a sample of plants. So now that we have collected our different plant species, just as before, we're going to tape them onto our board so that we can look at the different varieties of plant species and see if we find any difference in the open forest versus in the field versus the forest. So as we're looking at our samples from the forest, one of the things you'll notice is we're starting to see larger samples of leaves, and we're starting to see fewer samples of grass that's available in the forest. You'll also notice that if you go back and you look at the transect method, you'll see a lot of dead leaves that was on the forest itself. This dendritic material is very important because we're going to see that it increases the nutrient content of the soil and therefore increases the fertility of the soil. You'll notice that we don't see a lot of grass species in the forest because the canopy often will not allow enough sunlight to reach the floor of the forest, so that these species have a tougher time growing, as compared to the open field where we saw a lot of different grasses in the open field. So these are some ways in which we can observe the difference in diversity within these two aspects of the North Woods, and then talk about why would we see those different plant species in one versus the other.